Hi everyone, you are in the case study regarding the biochemical techniques to determine COVID-19 infection, which has been affecting the world population in the past few years. Let's see how biochemistry principles are applied to these techniques. COVID-19 is currently well known as an infectious disease caused by a severe acute respiratory syndrome coronavirus 2 virus or SARS-CoV-2 virus. Its emergence began in 2019. That's why its name is 19. The pandemic affects all people around the world with severe pneumonia, which is a cause to death. Screening and vaccination are primary regulations around the world. Our case study today is about a 45 years old woman who seemed well. She, for some reason, underwent a nasopharyngeal swab for SARS-CoV-2 virus screening by the antigen test kit known as ATK and the reverse transcription polymerase chain reaction known as RT-PCR. She had three vaccinations of the dead virus, the DNA, and the mRNA vaccines. Finally, her screening results were negative for the ATK but positive for the RT-PCR. Was such a confusing report. Before we have a look through the key learning point, let's understand the SARS-CoV-2 virus component. SARS-CoV-2 virus belongs to the coronavirus family, of which the genetic material is RNA, so-called RNA virus, wrapped around with the protein known as nucleocapsid or N protein. The whole structure is covered by an envelope made from phospholipid, where three key membrane proteins are embedded. Those are the spike protein or S protein, crucial for infection to the host cells, M and E proteins. Specifically, detection for the viral particle, RNA and nucleocapsid protein or N proteins are of interest. Of course, reverse transcription polymerase chain reaction or RT-PCR must be implemented for detection of the viral RNA. In the meantime, immunological assay must be applied for specific antigenic protein for the virus. This technique known as antigen test by a commercial kit so-called antigen test kit or ATK. Let's focus on how RT-PCR works to determine the viral RNA. RT-PCR is a combination of two molecular techniques, which are reverse transcription or RT and polymerase chain reaction or PCR. The RT is to convert the RNA of the virus to DNA by using a reverse transcriptase enzymes. Once the DNA is made, it is then used as a template for the PCR as required DNA, not RNA, as the template. The PCR is the technique to increase the number of the DNA fragment from the sample by imitating the replication process. Heating and cooling is used for denaturing the double strand of the DNA template. Specific primers, which are forward and reverse primers, are required to allow only certain fragments of DNA to replicate in each heating and cooling cycle. It also requires specific reporting molecules to detect the increasing number of the DNA fragment. In a cycle of PCR consisting of denaturation, annealing, and elongation phases, it theoretically doubled the amount of the template into over n fashions, when n is the number of the cycle proceeding. The product of 2 over n number is based on only a single copy of double-stranded DNA template is used. There are variety methods to determine the number of products produced from the PCR. This is one example using SYBR green, so-called cyber green, which is bind only to the double-stranded DNA to emit the fluorescence. At the end of elongation phase, cyber green bind to the PCR product and emit the fluorescent light. The fluorescent intensity is measured in every cycle after the end of the elongation phase for fluorescent intensity. The fluorescent intensity increases every cycle due to the duplication of the PCR product or the DNA template. When the results are reported, that is normally a graph plotted between the fluorescent intensity and the cycle number. It is a sigmoid curve. When the lower cycle number, the fluorescent intensity is low, reflecting the low copy number of the product. When it is suddenly increases the intensity, it exponentially increases. And when the first cycle at which the fluorescent intensity is detectable or higher than the baseline intensity, it is recognized as CT, which is standing for cycle of threshold. CT is very crucial for result interpretation. If you have two samples showing as a high CT and a low CT values, the sample show lower CT value contain the higher amount of the starter DNA template. For example, if the sample contains SARS-CoV-2 RNA and the sample does not contain SARS-CoV-2 RNA, the CT value of the sample containing SARS-CoV-2 RNA must be much lower than the CT value of the sample does not contain SARS-CoV-2 RNA. Let's move on to the detection of antigen by immunoassay, which is another popular technique to detect SARS-CoV-2 in the form of ATK. The principle behind this case is designed for the lateral flow of the reaction solution. The sample solution is added at one end next to be absorbed by the labeled antibody to detect SARS-CoV-2 antigens. This antibody is labeled with chromophore, which is an agent to generate color on the strip. The kit contains another two significant lines, which are test line and control line. When the sample solution contains SARS-CoV-2 antigen, it is captured by the labeled antibody, forming the complex during the flow. When this complex is reached the test line indicated by X, the complex is captured by the immobilized antibody recognizing SARS-CoV-2 antigen, so the color is developed in this line. The flow also taking the complex to the control line as well, indicated by Y, and the complex is also captured by the immobilized antibody recognizing the labeled antibodies. So the color is also developed here in this line. In contrast, when the sample solution does not contain SARS-CoV-2 antigen, no complex between the labeled antibodies and the SARS-CoV-2 antigen is formed. It is freely flowed to the test line without being captured by the immobilized antibody, which recognizes the SARS-CoV-2 antigen. So the color is not developed in this line. The flow continues taking the free labeled antibodies to the control line, and the free labeled antibody is captured by the immobilized antibody recognizing the label antibodies, so the color is only developed in this line. That's why two color lines develop mean positive detection of the SARS-CoV-2 antigen reflecting the infections, while one color line develop means negative detection of the SARS-CoV-2 antigen reflecting without infections. When comparing ATK and RT-PCR for determination of SARS-CoV-2, we found that both techniques are efficient to detect high to medium loads of the virus. However, low viral loads such as during the onset of the infection, ATK seem less sensitivity than that of RT-PCR. Apart from less sensitivity, ATK seem more advantageous in terms of cell operation and quick detection over the RT-PCR. Both of them could potentially report the false negative at similar rate of incidence. Prevention of SARS-CoV-2 infection, vaccination is strongly recommended. There are 4 types of vaccine against SARS-CoV-2 infection, so-called COVID-19 vaccine. DNA-based viral vector vaccine, attenuated or Q virus vaccine, mRNA vaccine, and protein subunit vaccine. This presentation only describes the first 3 types on how this vaccine activates our immune system against COVID-19. Queued and attenuated virus vaccine are developed by using the whole particle of the virus to process by killing it or just inhibiting the pathogenic activity. These vaccines can be engulfed by phagocytes and present the antigenic portion to the T white blood cells for developing the antibodies against the natural virus. So we have the immunity against this virus. In case of viral vectors or DNA vaccine, it is designed using non-pathogenic viral particle which must be genetically engineered to insert the SARS-CoV-2 genes into the vector genome and let the engineered virus do the job by infecting to the host cells. This type of virus sends the genome into host nucleus where the SARS-CoV-2 gene is transcribed there and exported to translate in the cytoplasm. The viral protein is then produced and expressed on the cell membrane of the host, which attracts the immune cell to destroy the cells and produce the antibody against it. Like DNA vaccine, mRNA vaccine proceeds similar mechanisms. The mRNA of the SARS-CoV-2 is produced and packed into the liposome system to protect the viral mRNA and deliver it. Liposome can fuse to any host cells to deliver the mRNA into the host cytoplasm, and the translation of the viral mRNA is triggered. Again, the viral protein is produced and expressed on the cell membrane of the host, which attracts the immune cells to destroy the cells and produce the antibody against it. So now we have the immunity against COVID-19.